Hey, welcome everybody to our second in the series of 2023 CBR seminars. Pleasure to be here. I just start off with just a couple of a few announcements. One, January 26th, when's that? Thursday? In a few days, from 4 until 6 up in the CBR pod, the fourth floor pod. There is a New Year's welcome. Food, whatever, social. So you're all welcome. Please come and extend that invite to other CBR members. To the Norman Bethune Symposium is April the 11th? Yes. Got it. Okay. So coming up soon. So students, trainees, staff, everybody start getting your abstracts together. Submit them quickly to Mira and to Parvin. Put that date aside, please, and spread the word. All right. 3, Vancouver Sun Run. There's the CBR Sun Run, 10 kilometers. Can you imagine? All right. So Ed Priso is going to lead that run, no doubt. He just doesn't know it. So there is a CBR team, but you have to get your registration in quickly, and you'll be partially reimbursed. Parvin's handing out the cash for that. And I will be there. And then on November the 16th of 2023, the Earl Davey Symposium. We're way ahead of the game. All right, so put that date aside. All right, so now on to the day. We had squirrely platelets last week. I think we're going to move more into the mouse and human realm today. So it's my pleasure to introduce Dr. Robert Campbell, or Robbie Campbell, who I met. You had already moved, I think, to Utah. So he did his undergraduate training in South Carolina and moved a little bit north to North Carolina to Chapel Hill, where he did his PhD and a short postdoc. I guess that was with Alyssa Wahlberg, who is well known in the hemostasis thrombosis world and here as well, and was extraordinarily prolific, but even more so when he moved to the University of Utah, where he did a postdoc there, and then was taken on as an assistant professor. He's done amazing work in platelets, particularly, but other areas of coagulation that are interesting to all of us. He's extraordinarily well published, well funded, does lots of teaching and speaking, and we're really happy that he came here from sun-filled Utah to rainy, dreary Vancouver. So thanks for coming, and we're looking forward to your talk.
Can you hear me? Yes, thank you so much for the invitation. This time of year, actually, there's never any sun in Utah either. Interestingly, I don't know, for those of you who probably have never visited Salt Lake, or if you have, we're situated between two mountains, and our pollution during the winter times can be worse than anywhere else in the world. So it's actually nice to come up and have fresh air and actually see some rain, since we rarely see rain in Utah. Again, thank you so much for the invitation. Today, I'm going to talk about some recent studies that we've published in our laboratory over the last couple of years, and feel free to interrupt me at any time if you have any questions. I'm happy to discuss any of our work. I'd like to first just to start out by thanking the individuals that have really done a majority of the work, and this includes Dr. Frederick DeNorm, who's a postdoc fellow in my laboratory, along with a large quantity of researchers here at the University of Utah, as well as in the University of Adelaide and Australia, as well as elsewhere in the United States. And also at the very end, I'll touch on some data that we generated with Eric Bouillard at the University of Laval in Quebec City. So platelets generally are in this discoidal shape here that become activated in a matter of seconds to form these large pseudopodia. And they can become activated when they see VWF, which is a plasma protein that helps them bind to a receptor on the platelet surface called GP1B alpha. They can be activated by soluble factors insoluble factors such as collagen and thrombin that causes initial stimulation and signaling. And then there's feedback systems like a thromboxane, release of ADP that can bind to P2Y12, which causes this shape change. And then they also have these other receptors on their surface that help allow them to adhere to other cells, but also to themselves. For the longest time, I think the field has really generally expected the platelets to be in this resting state, and then they become activated, and they basically just form a plug to prevent bleeding. But our lab likes to think about them in this role, in an immunohemostatic role. So platelets have these traditional dysfunctions such as adhesion, aggregation, degranulation. They can generate thromboxane, and they can play a role in clot retraction. But they also have these non-traditional, non-hemostatic functions. So they can sense other pathogens. They can signal to other cells. They have immunosurveillance properties. They can regulate their gene expression, mostly through their megakaryocyte, which is the parent cell. And what I'll show you today is they have this really wide dynamic transcriptome and proteome. They can splice RNAs and they can translate RNAs, which are very unique for a non-nucleated cell. This is from a recent review another postdoc in my lab wrote along with me, and really just show overview of the hemostatic function, non-hemostatic function, innate immune functions of platelets. So they have toll-like receptors. They can recognize viruses. They can generate cytokines that can then be released. They can recognize pathogens like viruses such as dengue and HIV. They can interact with infected red blood cells like plasmodium and malaria infections. They have these receptors such as Fc gamma, which can bind antibodies that are bound to pathogens. Some of these hemostatic receptors on their surface can also interact with bacteria. And they have all these really antimicrobial pathways and that people have generally become to accept that platelets do more than hemostatic functions. Our lab is really interested in thrombosis and certainly in the setting of infectious diseases. This is from a paper that a collaborator of mine, Matt Rondina, published many years ago, looking at venous thrombosis in the setting of sepsis. And when you took almost over 100 individuals that were septic and you scanned them for a clot, almost 40% of them had a clot. And these range from in the legs and in the arms, but generally speaking, individuals that are sick with infectious diseases are prone to having a clot or having a thrombotic complication. This sort of leads to what our lab mainly studies. So we're interested in how pathogens or inflammatory agonists change the genetic code or the transcriptome of megakaryocytes. And then the megakaryocyte, as it makes platelets, can then transfer these new RNAs and proteins into the budding platelet. The platelet can then also pick up signals from inflammatory pathogens or agonists, and then you have these new platelets that circulate with this altered genetic code, and then they can participate in these altered functions due to these changes in their RNA and their protein. So we became really interested in this in the setting of dengue infection, where we saw this protein called IFITM3, which is an interferon-induced transmembrane protein 3 that was upregulated in dengue patients. But we also saw this in the setting of influenza, and we also saw this in the setting of bacterial sepsis. And then you can see in healthy individuals, we saw very little transcriptome or RNA of IFITM3, but in dengue, influenza, and also in sepsis, we saw the significant upregulation of IFITM3. And we became interested in what this was doing. So IFITM3 is a well-conserved interferon-inducible gene. It has a YXXF motif that allows it to bind to clathrin. And it basically allows viral particles to come in, and IFITM3 basically blocks the ability of the virus to fuse with lysosomes and release the genetic or the virus into the cell. So it restricts viral replication of dengue and influenza by blocking its ability to interact with lysosomes. And it's well conserved in humans and mice. And we began to ask, well, why is this upregulated in viruses or in virus-infected patients? And we thought that maybe upregulation of IFITM3 and megakaryocytes and platelets would prevent viral infection. So we looked at this a little bit more in dengue patients. So in dengue patients, we saw a significant increase in the amount of interferon alpha in mild dengue patients, but also in more severe dengue patients. And this seemed to correlate with the increase in IFITM3 protein. So in our mild dengue patients, we saw a significant increase in IFITM3 or protein in platelets, but we also saw this in our severe dengue patients as well. And it sort of loosely correlated with interferon expression. So as you had more interferon expression, we saw that there was more IFITM3 in the platelet. We also saw something very similar in influenza-infected patients, and these were recruited from the University of Utah. So if we look at healthy patients, again, they had very little interferon, our influenza patients, they had more interferon, and this correlated with more IFITM3 protein in the platelets from influenza patients, but also more protein. And interestingly, this seemed to correlate in terms of survivors versus non-survivors. So if you followed septic influenza patients over the course of their ICU stay, and you looked at IFITM3 expression, individuals that had no increase in IFITM3 tended to not survive the infection, whereas individuals that actually had an IFITM3 response tended to do better. And again, this is a small number of patients, but it seemed to suggest that IFITM3 upregulation seemed to regulate potentially infection in these patients. So we went to a model system where we could actually study this from an in vitro perspective, and we did this in a CD34-derived megakaryocyte system where basically we were able to differentiate hematopoietic stem cells into megakaryocytes, which are sort of the precursor to the platelet. And we infected those with dengue virus. And you can see when you either gave them mock or dengue, we saw a significant increase in the amount of IFITM3 expression on their surface. And this is quantified here, where mock cells had very little IFITM3, but when you gave them dengue virus, dengue 2 virus, we saw this significant increase in the amount of IFITM3 expression. And interestingly, when we looked at this from an ICC perspective, here on our left is our mock, we saw very little IFITM3 expression, which is in our red. But when we gave dengue virus, we noticed that several of the megakaryocytes would become infected with dengue or were dengue positive. But interestingly, those cells tended to be IFITM3 negative, whereas the surrounding cells seemed to be very, very robustly expressing IFITM3, but tended to be dengue negative, suggesting that maybe the IFITM3 upregulation was preventing dengue from infecting these cells. So we went ahead and dissected this a little bit further. We took megakaryocytes and gave them interferon, so sort of a pharmacologic manipulation. And as we gave them more interferon and we looked at infection, we saw our wild type cells were infected. Our vehicle infected cells were also infected. But when we treated the cells with interferon, it blocked the ability for dengue to infect the cells by ICC imaging. We then looked at manipulating the cells genetically. And how did we do this? So we overexpressed MEGA1s with IFITM3. And you can see up here, when we overexpressed IFITM3, we saw this robust increase in IFITM3 expression. And then we went ahead and infected the cells. And our wild type cells became infected or became dengue positive. But when you overexpressed IFITM3 in the megakaryocytes, this seemed to block the ability for the cells to take up virus or be infected by virus. The converse of this is downregulating IFITM3, and we did this through shRNA. And in this instance, we were able to induce IFITM3 expression with interferon. And when we did the same thing where we had the shRNAs against IFITM3, we saw a significant reduction in IFITM3 responses. And then we went ahead and looked at the ability of those cells to be infected. So in our wild type cells, we saw some level of infection, but when we were able to block IFITM3 with these shRNAs, we saw an increase in infection, again, suggesting that IFITM3 was regulating infection, dengue infection, and CD34 or megakaryocytes and platelets. So as I mentioned earlier, this protein was up in dengue individuals. It was also up in influenza patients, but it was also up in bacterial sepsis patients, which is sort of an interesting phenomenon because in these patients, they had had known diagnosis of bacterial infections, no viral infections. So in platelets and megakaryocytes, IFITM3 is upregulated in influenza, dengue, I didn't show you today, but also in COVID, and it seems to augment these antiviral activities. In platelets, hemostatic cargo can be endocytosed through clathrin-mediated endocytosis, such as fibrinogen. And this can promote cell activation and it can promote hemostasis, but it can also promote thrombosis. And we asked whether or not IFITM3, since it's regulating clathrin-mediated endocytosis of viral particles, if it could regulate endocytosis of other hemostatic cargo and platelets, such as fibrinogen. And this sort of diagram looks like this. So fibrinogen can be endocytosed through clathrin-mediated endocytosis. It gets packaged into endosomes, and then it gets merged into alpha granules where VWF is. And then once the platelet gets activated, it gets exocytosed out. And why is this important? So you can form platelet aggregates. The platelets can then interact with other leukocytes, and they can also interact with the endothelium cells when they have increased fibrinogen or when they are activated and they're surrounded by fibrinogen. So we asked whether or not IFITM3 was altering other forms of platelet function, potentially by upregulating fibrinogen endocytosis, and that's sort of the hypothesis we came up with. So upregulation of IFITM3 and platelets during sepsis induces clathrin-mediated endocytosis of hemostatic cargo, which can then alter how the platelets become activated and thrombotic complications. So we did this first by looking at whether or not interferons were upregulated in septic patients, and indeed they were. So in our healthy controls, we saw very little levels of interferon alpha, but we saw a significant increase in our sepsis population. We also saw that there was a significant increase in sepsis platelet IFITM3 in these septic patients, and it was quantified here. So we saw, and again, on our healthy, very little level of IFITM3, that significantly was increased in our septic patient population. And we also saw in our septic patients that they had increased fibrinogen inside their platelets, suggesting that maybe IFITM3 was regulating fibrinogen from being endocytosis into the platelet and or the megakaryocyte. We then looked at platelet activation in the form of aggregation, and we looked at this specifically with collagen, but we've also looked at this with ADP. And we saw in our healthy controls under low levels of agonists such as collagen or ADP, we saw basal activation, aggregation. But when we activated with collagen, and we looked at this in sepsis patients, and again, a similar thing was seen with ADP, we saw hyperreactivity that is quantified here, again, suggesting that upregulation of IFITM3 is associated with increased fibrinogen endocytosis, but also platelet hyperreactivity. We next turned to a knockout model, and unfortunately for the paper that we published and for the talk, this was a global knockout where it's not platelet specific, and IFITMs are globally found on the same region of the mouse chromosome, and in this mouse that actually knocked out all the IFITMs, but specifically the one that we're looking at, IFITM3 is the only IFITM that has that YXXF motif, which is important in clathrin-mediated cytosis. But when you gave the mouse interferon through IP injections, we saw a significant increase in the amount of IFITM3, whereas in our knockout animals, if you gave them interferons, we saw no change in IFITM3 in the platelet.
Bobby?
Yes.
Just a quickie. On that note, is IFITM3 expressed on endothelial cells?
Yes, it's ubiquitously.
And is it upregulated?
Yes.
By interferon?
Yes.
And does it have effects as far as you know?
We have not looked, but it could. In this setting, though, these are washed platelets, so what we're looking at specifically is a platelet-mediated effect. But from an in vivo perspective, it is possible that it is having an effect from other cell type. Yeah, that's entirely possible. We have made a platelet-specific knockout, and we see similar things. But when we started the project, it just took us some time to make the platelet-specific knockout. But in this model, we took wild type and knockout animals, and we put them under a cecal ligation puncture model, which is a polymicrobial sepsis model, where you ligate the cecum and put feces into the peritoneum, causing a peritonitis model. And we saw a very similar phenotype in our septic mice as we did in our human septic patients. So shams, we were able to aggregate. Sepsis animals, and wild type animals, we saw a significant increase in aggregation. But in our knockout animals, we saw no change in aggregation in wild type or CLP animals, and that's quantified over there on the right-hand side. We know that this is interferon dependent because if we add an anti-interferon antibody, it blocks the response. So these are in wild type animals. Again, wild type animals aggregate in the presence of CLP or under CLP conditions. We saw a significant increase in aggregation. If we give the animals an anti-interferon alpha receptor antibody, it basically blunts the response, and this can be shown here. Again, we see an increase in aggregation that's blunted by the effect of anti-interferon receptor antibody, and we can also blunt the ability for IFITM3 to go up, suggesting, again, this is an interferon-mediated response and potentially an IFITM3 specific response. We then looked at fibrinogen endocytosis in these mice. Again, this is a more sterile inflammatory response where we were giving them interferon alpha in our wild type animals versus our knockout animals. In our wild type animals, they have baseline levels of fibrinogen that significantly increase in the presence of interferon. But in our knockout animals, where we are looking at platelet fibrinogen, we saw no change in the amount of fibrinogen that was there when IFITMs were absent. We can also look at this in an in vitro cultural system where we culture megakaryocytes from the IFITM deficient animals, and we can look at labeled fibrinogen uptake, which is in the magenta. So under wild type conditions, we saw fibrinogen that was endocytosed. If you give them interferon in the culture system, we saw a significant increase in the amount of fibrinogen that was present in the megakaryocyte. In our knockout animals, in the absence or presence of fibrinogen or interferon, we saw no change in fibrinogen endocytosis. The megs were able to take up fibrinogen, but this enhancement was not present in the absence of interferon and IFITM3. We also can look at this from the setting of, again, sterile inflammation, where we give mice interferon in our wild type on our knockout. So in our wild type situation where we give them interferon, we saw this significant increase in platelet aggregation in response to ADP, but also in the collagen. But when we look at our knockout animals and we give them interferon, we saw very little enhancement in the amount of aggregation that was occurring, again, suggesting interferons and IFITM3 are regulating the ability of the platelets to become hyperreactive. And finally, we looked at two models of thrombosis. One was a ferric chloride model, and then another one is a pulmonary embolism model. And in the ferric chloride model, we basically expose the carotid artery and put a little bit of ferric chloride on there for a short period of time and remove it, and then we monitor blood flow and we can look at occlusion. And in this model, we saw very little occlusion in our wild type animals, but when we give our wild type animals that have IFITM3 there interferon, we saw a significant shortening of the occlusion time, suggesting that they are more prone to thrombosis. But in our knockout animals, in the presence or absence of interferon, if they don't have IFITMs there, we saw no difference in thrombosis. And a very similar outcome was observed in our PE models, where in our wild type animals, interferon was a significantly increased the amount of death associated with the PE model, where in our wild type, or in our knockout animals, in the presence or absence of interferon, we saw no change in thrombosis. So this suggests really that IFITMs were regulating thrombotic complications in mice in the setting of interferon, but also in the setting of thrombosis. So how do we know with IFITM3, or why do we think it's IFITM3? Well, a couple reasons. So we took those cells that we overexpress IFITM3 again, and we can see that it's upregulated here. And we looked at fibrinogen endocytosis ex vivo in a culture system, and we saw a significant increase when we only expressed IFITM3, suggesting that IFITM3 was specifically mediating this response in megakaryocytes. We also knocked IFITM3 down. So in this instance, again, we give the megakaryocytes interferon. We induce an increase in IFITM3. We also induce increases of other interferon IFITMs. But we knock down specifically IFITM3, which is shown here. And when we do that, we see an increase in the amount of fibrinogen endocytosis in our wild type controls where we give them interferon, whereas in our knockout animals, our knockout where we knock down IFITM3 and we give them interferon, we saw no change in the fibrinogen endocytosis, suggesting that, again, IFITM3 was specifically mediating this response. So how might it be doing this? Well, IFITM3, again, has this YXXF motif that is associates with clathrin. And when we pull down IFITM3 and we look at clathrin, we see clathrins there, but we also see alpha-2b. So alpha-2b is the major fibrinogen receptor on the platelet or megakaryocyte surface, suggesting that fibrinogen, or alpha-2b, clathrin, and IFITM3 form a complex. And potentially that's how IFITM3 is able to regulate fibrinogen endocytosis. Also, very interestingly, IFITM3 associates with lipid rafts, and we saw a change in the lipid rafts of platelets when IFITM3 was present. So here's our megakaryocytes that don't have IFITM3 overexpressed. We can see IFITM3s in the non-lipid raft fractions, sort of in the cytoplasmic fractions. We saw alpha-2b was present there, and clathrin was there. We measured LAT as a marker of lipid rafts, and we saw very few proteins associated in the lipid raft fractions. But when we overexpress IFITM3, we see this large shift of IFITM3 over into the lipid raft fraction. We also saw a shift of alpha 2B into the lipid raft fraction. And we also saw this shift in clathrin, suggesting that IFITM3 was changing the localization of these proteins to help facilitate clathrin-mediated endocytosis. So this is in a culture system. Do we see this also in a patient population? And indeed we do. So these are platelets from healthy donors or sepsis patients. And again, we're looking at lipid raft fractions versus non-lipid raft fractions. So we saw very little IFITM3 in the non-lipid raft fractions of our healthy donors. Alpha 2B seems to be there as well as clathrin. And here's again our lipid raft marker and our lipid raft fraction. But in our sepsis patient population, we saw this increase in IFITM3 and this shift over into the lipid rafts. And we also saw this shift of alpha 2B and clathrin as well. And this, as can be shown here, where again our lipid raft marker. So again, suggesting that IFITM3 was moving the location of alpha 2B and clathrin more towards lipid rafts, where it could facilitate clathrin-mediated endocytosis fibrinogen. So that's sort of the first part of my talk, looking at how infection can alter the platelet transcriptome, can alter the proteome as well, specifically with IFITM3, but there's also thousands of other genes that change that I don't have time to talk about today. So in MEGs and platelets, there's an IFITM3 antiviral property that happens that can help fight infection, and this may have a role in preventing megakaryocytes from dying and the associated thrombocytopenia associated with many viral infections. But IFITM3 can also alter how the platelets are function during sepsis and potentially also during antiviral or viral infections as well. So the other thing my lab is very interested in is understanding how platelets interact with other cells. And the other cell that we're really interested in right now is neutrophils. So in general, platelets can interact through expressing P-selectin on their surface to bind to PSGL ligand on the neutrophil surface. It can also interact through this MAC1-GB1B alpha complex. And the platelet can then release things to affect neutrophil function, but the neutrophil can also release things to affect platelet function. And this can result in phagocytosis, oxygen or ROS generation, leukotrienes, tissue factor-mediated coagulation. It can cause the neutrophil to induce tissue repair. But one of the things that we've become really interested in are these things called NETs or neutrophil extracellular traps. So why are we really interested in the platelet and the neutrophil in this setting? So there's good data out there to suggest that if you have a stroke and the level of neutrophils can correlate with those outcomes. So as you have more neutrophils, you have worse outcomes in stroke. And this is this is quartile, the neutrophil level in the blood. And this is based on modified Rankin score or neurologic function. But it's also associated with death. So the more neutrophils that you have in your blood and you have a stroke, you're more likely to have a mortality phenotype. It's also been shown early on in the disease process. So when you're admitted into the hospital, if you have more neutrophils, you tend to have a higher modified Rankin score. So again, more neurologic dysfunction. And this is seen again at day 1 as well. So neutrophil or neutrophilia is associated with worse stroke outcomes. So where do platelets and neutrophils sort of come into this world? So besides P-selectin, platelet PS or phosphatidylserine can also regulate these interactions. And this is work by Ben Kyle's group in the University of Adelaide, where they took PS-positive platelets and they perfused neutrophils over them, or they took PS-negative platelets and perfused neutrophils over them. And you see, and when you have PS-positive platelets, you form these large macroaggregates that are not present when you have PS-negative platelets. And you can see this more readily here under ICC. So PS-positive platelets in green, you form these large macroaggregates. If you have PS-negative platelets or microparticles, you can see some binding of the platelets to the neutrophils, but very few macroaggregates. So it's suggesting that platelet PS is a critical regulator of neutrophil platelet interactions. So how do you get platelets to make PS? So there are multiple different ways. The way that we like to think about it is there's thrombin and collagen, or so you have this dual signal. You can also have ROS or hypoxia, but in general, what happens is this causes an increase in intracellular calcium. That calcium then fluxes into the mitochondria and then out through a molecule called C5D when this mitochondrial transition membrane pore. And this calcium efflux then activates these scramblases to induce phosphatidylserine expression. So when you knock out platelet-specific C5D, again, this molecule here that activates the releases of calcium and that results in the scramblase activation, and you activate platelets, either wild type platelets express PS, but when you activate these C50 deficient platelets, you get a significant reduction in PS. So we asked whether or not platelet PS or procoagulant platelets are key regulators of ischemic stroke and through the recruitment of neutrophils. And to do this, we use a transient middle cerebral artery ischemia model where we take a filament that's shown right here, feed it through the carotid artery up into the circle of Willis, and then put this filament right at where the MCA is, and it blocks blood flow for a certain period of time. And when you do that, you end up with this infarct core, which is shown right here. Then you can remove the filament and let the animal have reperfusion for some period of time. And generally speaking, this is for about for a 24-hour period. And this allows for the infarct to grow. And when we did this in our platelet-specific C50 knockout mice, we saw a protective effect. So in our wild-type animals, we saw normal stroke right here, which is quantified here in terms of the volume of the stroke. But when you knock out platelet PS, the mice are protected. You just have that infarct core that's there. You don't have this reperfusion injury. And this was partly mediated through the fact that you have a decrease of cerebral blood flow in our wild type animals that was not observed in our platelet C50 knockout animals. So we can measure cerebral blood flow at baseline during ischemia, and then at reperfusion it goes up to about 50%. And in our knockout animals, it stays basically the same over a 24-hour period. But interestingly, in our C50 knockout animals, our C50 wild type animals, we saw this significant decrease in blood flow, suggesting that the reason why these animals have increased brain injury is because they have reduced cerebral blood flow that was not present in our C50 knockout mice. We looked at platelet monocyte aggregates, circulating platelet monocyte aggregates after stroke, and this was no different in our wild type, our knockout animals. But interestingly, there was a significant reduction in our platelet neutrophil aggregates in our C50 knockout mice, suggesting that maybe platelet neutrophil aggregates were having an effect. And we looked at the platelets that were bound to the neutrophils and looked at PS positive platelets on them. We again saw a significant reduction in the amount of PS positive platelets associated with those platelet neutrophil aggregates, suggesting that again, platelet neutrophil aggregates and potentially PS platelet-filled neutrophil aggregates were driving this phenotype, and potentially the lack of them was resulting in this protective phenotype. In vitro, we were able to look at platelets and their ability to interact with the neutrophils. So here are our wild-type platelets, here are our knockout platelets, and we dual stimulated them. And you can notice in our wild-type platelets, when we dual stimulate, you see these vacuolated platelets that are not present in our knockout animals, suggesting that, again, that these animals are incapable of flipping their PS. And when you put neutrophils with them, you can see these large neutrophils interact with these vacuolated platelets. And you can see here that they preferentially are sort of forming these multiple tethers to the neutrophils that are not really present here in our knockout animals. You can see that you form these neutrophil platelet aggregates, but you're not forming these multiple tethered neutrophils that are bound by these procoagulant or necrotic platelets. We then looked at neutrophil recruitment to the brain in these C50 deficient animals, and we saw a significant reduction. So again, it seems like when you knock out the ability of the platelet to form PS and you give them a stroke, you see a reduction in the amount of neutrophils that are being recruited to the brain, that are where you see a significant amount of neutrophils that are brought to the brain in our wild type animals. And then we then did ICC, and we looked at where the neutrophils are. And you can see in our wild type animals in MPO positive, which is a stain for neutrophils, we saw neutrophils that are there that are present with green platelets, which are GP1B alpha. And you can see in these two images here that there are platelets and neutrophils that are present. And in our knockout animals, we saw some platelets there, but very few neutrophils there, again, suggesting that this recruitment of neutrophils to the brain after stroke when you block PS, platelet PS, is reduced. So why may this be important in stroke patients or in stroke? So interestingly, we did RNA-seq stroke patients, and we looked at these genes, MCUR1, MCURB, which are regulators of this calcium flux, along with this TMEMF16 and calpain, which regulate PS expression. And interestingly, in stroke patients, this positive regulator of mitochondrial calcium and this negative regulator of calcium are changed. So MCR1, which is this positive regulator, is significantly upregulated in stroke patients. So you would think they would have more calcium being influxed into the mitochondria. and they have this negative regulator that goes down. So in theory, that they would again have another reason for more calcium to be increased in the mitochondria. And then if you look at these two PS regulators, TMEM-F16 and calpain, we saw a significant increase, suggesting again that stroke patients may be prone to flip their PS when they're activated. And this is work that is going on in our lab currently, trying to understand how PS and calcium in platelet neutrophil interactions are altered in stroke patients. So there are many reasons why people have targeted platelets, as I've shown you, that they play a role in stroke, but some people have tried to target neutrophils as the other player in ischemia reperfusion injury. And there are limitations with current anti-neutrophil strategies. They assume that neutrophil extravasation or neutrophil recruitment and then going into the brain is the key mediator. And they're also targeting the ability of the neutrophil to interact with the endothelium. And they're targeting classic neutrophil interactions. And the reasons why these have not worked previously is because they have generally targeted these functions. And in clinical trials where they've targeted anti-ICAM1, for example, they've actually shown no benefit or actually even worse outcomes. So we like to think about something called a neutrophil extracellular trap, which are released from neutrophils when they become activated. And evolutionarily, they have played a role in trapping bacteria, which you can see here. These long strands of DNA are trapping bacteria that prevent them from spreading in the body. However, neutrophils can also extrude these and trap platelets and activate the contact pathway. So we think that neutrophil extracellular traps play more of a role in thrombosis than actually in bacterial killing. And we thought about their role in ischemia reperfusion injury, where platelet neutrophil interactions are important in inducing these neutrophil extracellular traps to induce immunothrombosis and neurotoxicity. So the first question we asked was whether or not NETs were actually present in human stroke patients, and indeed they are. So we were able to get brains from stroke patients that have passed away, and we stained for neutrophil elastase, MPO, H3-CYT, which is a neutrophil-specific marker, and DNA. And you can see here and here that they're clearly neutrophils that are present. And you can see basically here, but it's also blown up here, that there's these extracellular strands of H3-CYT, suggesting that these neutrophils have undergone netosis. And these are sort of a 3D rendering of this as well, that they're positive neutrophils that have been recruited to the brain, and that they're positive for H3-CYT, suggesting that they have undergone netosis or they're in the process of undergoing netosis. So this is just one example. Here's another example where, again, here's sort of a blood vessel that's littered with neutrophils in this patient that had a stroke. And most of these neutrophils are positive for H3-CYT, suggesting that they're either undergoing netosis or they have undergone netosis. And over here is an example of neutrophils that are interacting with platelets. So in the stroke brains, we see neutrophils that are interacting with platelets that appear to have undergone netosis. So what happens in the circulation? Can we see markers of immunothrombosis? And we can. So if you look at D-dimer, which is a marker of fibrinolysis that's increased in stroke patients, we see increased PF4, which is a marker of activated platelets that are increased in stroke patients. We see increased calprotectin, which is a marker of activated neutrophils in platelets. So markers of immunothrombosis are increased in stroke patients, but also NETs are as well. So if we look at H3-CYT, so circulating H3-CYT, we see a significant increase in H3-CYT in stroke patients. But we also see a significant increase in the amount of MPO DNA ELISA, so MPO DNA. So that's a marker of NETs where MPO is complex to DNA, and we can measure this with an ELISA. Interestingly, as well, as we see an increase in DNase or decrease in DNase activity. So DNases will break the NETs apart, suggesting that maybe in stroke patients you have an increase in NETs, but also a decrease in the ability for them to be degraded, and that's something that we're following up in our lab. But when you look at these markers and you sort of correlate them with stroke severity or sort of disability score, we only showed that MPO and H3-CYT correlate with disability score, suggesting that NET markers and none of the other markers associated with immunothrombosis correlate with stroke outcomes, only this MPO DNA complex. So again, suggesting that NETs are important in the pathophysiology of ischemia reperfusion injury. We saw very similar things in our mouse brain and our mouse model. So again, here's a picture of a mouse brain after stroke, and we can see, again, in CD41 positive platelets, MPO positive neutrophils, and those neutrophils tended to be H3 CYP positive, suggesting that nets and platelets are present in the brain after ischemia and reperfusion injury in our mouse model. So what may the platelets be doing? So I've talked a little bit about how the platelets interact with the neutrophil, but again, the platelets release a ton of things that can activate the neutrophils, and we sort of honed in on one protein that we became interested in called HMGB1, which is a DAMP that has been shown to play a role in net formation in the setting of venous thrombosis. And we first looked at this, again, by looking at platelet-neutrophil interactions, and we saw a significant increase in the number of platelet-neutrophil interactions in our stroke patients compared to our healthy controls. And when we looked at HMGB1 positive platelets, we saw the significant increase in the amount of platelets that stained for HMGB1, suggesting that platelets were becoming activated and expressing HMGB1 on their surface. We saw this by a percentage, but also by the MFI, so the amount of HMGB1 that was there. And we also looked at this in the plasma, and we saw a significant increase in the amount of plasma HMGB1. HMGB1, again, is a DAMP, so it's capable that it's coming from other cells. But in general, we saw a significant increase in our platelets, which correlated with the amount that we saw in the plasma as well. If we look at the clots that were retrieved from stroke patients, we also saw a significant increase, or we saw HMGB1 that was present along with platelets and neutrophils, suggesting that HMGB1 platelets and neutrophils were associated with the thrombi and the stroke. So what may HMGB1 doing in this model? So HMGB1 goes up in our mouse model, and it correlates not quite nicely with NET. So as we give a mouse a stroke, we saw this significant increase in HMGB1. And we saw the significant increase in the amount of nets that were present. If we make the mice thrombocytopenic, so we deplete the platelets, we see this drop in the amount of HMGB1. And interestingly, we also see a drop in the amount of MPO DNA complexes, suggesting that the platelets themselves were contributing to HMGB1, but also contributing to the amount of nets that were being released in our mouse model of stroke. And this is seen as well as in the stroke volume. So if we have our control mice, we have a nice stroke. We can calculate the volume here. But when we make the mice thrombocytopenic, we saw the significant decrease in the amount of brain injury. So what happens if we deplete the other end of the spectrum, so neutrophils? So if we deplete neutrophils, we saw no change in the amount of HMGB1 that was present, suggesting that the neutrophils were not contributing to the HMGB1 that's released in this model. But without neutrophils, you don't see any nets, which is consistent here. So we saw nets in our wild type animals. If we deplete neutrophils, we saw no increase in the amount of MPO DNA complexes. And we saw protection though. So even though we have an increase in HMGB1 here, we saw a decrease in the amount of infarct. So again, our wild type, we have a nice stroke, and our neutrophil depleted, we saw the significant decrease in the amount of stroke injury. So what happens if we add back recombinant HMGB1 in our thrombocytopenic mice to sort of dissect the role of what the platelets are doing? So again, we have this stroke that's here, that's decreased when we make the mice thrombocytopenic, again through an antibody depletion. And when we give the mice recombinant HMGB1 that are thrombocytopenic, we can increase the amount of nets that are released. We can increase the infarct volumes, again suggesting that recombinant, or we're suggesting HMGB1 released by platelets are inducing this response. So that's sort of a little bit of hand waving to get to the genetic component of what's really going on. So we were able to make a platelet-specific HMGB1 knockout animal, which is shown here with the flox, and we crossed it to a PF4 Cree, which knocks HMGB1 out specifically in platelets. And we measured HMGB1 levels after stroke in these mice. And on our wild type animals, we saw HMGB1 levels go up that were significantly decreased in our platelet-specific HMGB1 knockout, suggesting that HMGB1 from platelets was the main regulator of this increase in plasma HMGB1 after stroke. We then went ahead and looked at nets that are being formed in this model. And again, we saw nets in our wild-type animals that were significantly decreased in our HMGB1 platelet-specific knockout, again suggesting that the HMGB1 that's being released by the platelets is contributing to the nets that are being formed. And then we went ahead and looked at the infarct volume or injury. In our wild type animals, we saw stroke that is quantified here. But when we knocked out HMGB1 specifically and in our platelets, we saw a protective effect in our stroke model, suggesting that platelet HMGB1 was specifically regulating NETs and ischemic stroke injury in this model. People have looked at blocking HMGB1 previously, but there is a protective effect of HMGB1 that is seen later in the stroke injury. So targeting HMGB1 may not be the best marker. So we thought about how do you inhibit NET formation. And again, the main question is, if we inhibited NET formation, would we protect our animals in our stroke model? And how do we do this? So a collaborator of mine made an observation about 10 years ago that if you take neonatal neutrophils, so neutrophils isolated from cord blood, they don't make NETs. You isolate the NETs or neutrophils from neonates 3 days after life, they make some more NETs. But by day 14, day 21 of life, day 28, excuse me, they make NETs. And this is quantified here, suggesting that either the neutrophils from neonates are immature or suggesting that there's some plasma factor out there that is preventing the neutrophils from making NETs. And to make the story short, there's a cord blood factor out there that's actually the N-terminal or C-terminal end of alpha-1 antitrypsin. And there's also a cancer motif called CRISP, but we named the NIF, which is neonatal net inhibitory factor. And again, it's the C-terminal end of alpha-1 antitrypsin. And you can see when you use an antibody against alpha-1 antitrypsin, or sorry, the C-terminal end of alpha-1 antitrypsin, and we basically purified this. We see NIF is present here, not in adults, in our cord blood, not adults, cord blood, not adults. So this small fragment is found in cord blood, but not in adults. And when you take this fragment, it blocks NET. So here's our control neutrophils. If we stimulate those with LPS, which is a DAMP or PAMP that can activate toll-like receptor 4 and induce NETs, we see NETs. If we take alpha-1 antitrypsin, so the full-length protein, and add LPS, you make NETs. But when you just give them the fragment of the C-terminal end and add LPS, they don't make NETs. So this endogenous C-terminal end of alpha-1 antitrypsin, we decided maybe this would be a good therapeutic option in this model. So again, we used this 1 hour of ischemia, 24 hours of reperfusion, and we looked at giving NIF here at 1 hour before and then 1 hour after. So this is more of a prophylactic model. And then we measured multiple things such as infarct size, neurologic, and motor function with these two tests, and then neutrophil recruitment. So if you give them the scramble peptide, which is the same amino acids, just in a random sequence, we were able to get a stroke that's very similar to what normal individuals or normal mice would be. But when you give them NIF, again, which blocks NET formation, we saw the significant decrease in the amount of infarct volume that was present. We also saw a better neurologic function, so a lower score is better with this Peterson test, and we saw an improved motor function, so an increase here in our grip test, suggesting that besides improving infarct volume, we were able to improve outcomes such as neurologic and motor function. We looked at the ability of the neutrophils to be recruited, and while we saw neutrophils that were net positive, which are these stars in our scramble mice, we saw neutrophils that were recruited in our NIF mice, NIF-treated mice, but we didn't see any NETs. So it suggested that neutrophils were being recruited, but they just weren't capable of making NETs. And if we look at the amount of MPO DNA in the plasma of these animals, when we give them NIF, we saw a significant reduction in the amount of NETs that were present in the plasma of these animals. So we next looked at giving this from a therapeutic perspective, because giving anything to an animal before or human before the injury is not really clinically relevant. So in this model, the therapeutic model, we gave NIF and scramble at 1 hour after the ischemia or 2 hours after the ischemia. And we measured those same sort of things, infarct function and then neurologic and motor function. Interestingly, in our 1 hour, we saw protection. So in our scramble mice, we saw stroke. In our NIF-treated mice, again, 1 hour after the ischemia, we saw a protective effect. If we give the molecule 2 hours after the stroke, we saw no protective effect, suggesting that there's a time window that's very sensitive, at least in this model. And I'll get towards that at the very end, what we may think is going on. But again, if we give this therapeutically, we can prevent stroke, at least at 1 hour, but not at 2 hours. So how about long-term? So in this model, we gave the peptide either 1 hour, so more of a prophylactic, or a therapeutic model an hour after ischemia reperfusion, and looked at the long-term effects, again measuring more motor function and neurologic function. So we did this by using this modified neurologic scoring system, which looks at behavioral functions, and we also measured this using a rotor rod, which is basically how well an animal can keep balance on a spinning wheel. And our blue is our scramble, and we see that in scramble mice, they have pretty poor neurologic function that maybe tends to go down over time, and they have poor ability to stand on the rotor rod or to stay on a moving wheel at 1 week, 2 week, and 3 weeks after the stroke. But in our NIF prophylactic, so again, giving this before the injury, or in our therapeutics, giving it an hour after the injury, we were able to see pretty robust protection in our neurologic function that was prolonged over a 3-week period, but also in our motor function, again, our prophylactic and our therapeutic, that the mice had improved motor function and neurologic function that was durable over a 3-week period. So why might we be able to give this at a 1 hour but not at 2 hours? And that has to do with something called, what we think called no reflow phenomenon. So this is a normal rabbit brain, and if you give it even 5 minutes of ischemia, you can begin to see, and then perfuse a dye, you can begin to see white patches where blood flow cannot get to even after 5 minutes of ischemia. And this is more prevalent even at 7.5 minutes or even 30 or 15 minutes. So even after 15 minutes of ischemia, and then you allow them to reperfuse for 30 minutes with this dye, there are large areas of the brain that do not get blood flow. There's clinical evidence for non-reflow phenomenon. So this is in patients where regions of no flow were identified in over almost a quarter of 130 patients. And then in this other study, they showed that tissue optimal reperfusion was strongly associated with good functional outcomes. So no reflow phenomenon is there in humans as well. So I think what I've driven today is that platelet adhesion drives brain injury. Platelet neutrophil interactions also drive brain injury, but neutrophils also regulate blood flow. And this is from our paper where we depleted neutrophils and we looked at cerebral blood flow. And again, we see blood flow that goes down under ischemia, that goes back up during the reperfusion period. And when we have neutrophils present, again, it goes down slowly. It's very similar to where we knock out platelet PS. But when we knock out neutrophils, we have very stable blood flow, suggesting that neutrophils are mediating this cerebral reperfusion injury. So what may be going on? So this is where Dr. Eric Bouillard and his graduate student Audrey LaRoche helped us do some 2-photon microscopy using a triple transgenic mouse that expresses neutrophils that are red and platelets that are green. And we were able to do 2-photon microscopy. So the 2-photon allows us to look in this area of risk, but not in this ischemic lesion. So we were able to look at the very cortex of the brain. And we can see here again, the platelets are in green, the neutrophils are in red, and we can look at, if it's going to work, yep. So we can see the green platelets sort of rolling by and neutrophils sort of scrolling along the endothelial cells. And we can sort of get an idea of what may be going on hours after the ischemia injury during the reperfusion period. So that was an example of what blood flow looks like after a stroke, but this is what normal blood flow would look like in a normal mouse. So you see very few neutrophils that are buzzing around. You see some platelets that are floating around, but you have very robust blood flow in the brain in a normal mouse. But even 1 hour ischemia, you can begin to see that it's really gummed up. You can clearly see individual platelets. You can see some areas of areas where it's completely clogged and a neutrophil is stuck. And then over here, you can sort of see this clot that's there, and you can see cells traveling this way, and it just sort of gums up the whole system. And over here, this is even 2 hours after reperfusion. You can see over here in all these small vessels, there's very little blood flow that's going on, and you can see it slowing down, and then all of a sudden you see blood flowing this way, and then it will flow back the other way here in a second because it gets clogged. It's gonna, yeah. How's it going to go? But anyways, even shortly after reperfusion injury, you can already begin to see that the blood vessels in the brain are very gummed up. And we also see these strange things like elongated neutrophils, and you can see up here in a second, so a lot of platelets that are clumping along a bit, and these neutrophils that are more look like pancakes than actually round cells that are adhering to the endothelium up there. And then we also see these things that we called cloudy neutrophils, and you can sort of see like a haze of neutrophil there and there. And potentially, these have netted. And we're beginning to look, and there's a clear one right there. We're beginning to look at those and see what's going on. So net markers are increased in ischemic stroke patients and in mice, and the nets appear to correlate with stroke outcomes, and we think targeting nets with NIF may improve stroke outcomes, and we're now trying to understand this no reflow phenomenon and sort of the therapeutic and mechanistic windows that we can look at to prevent stroke injury. And with that, I will thank you for your time and happy to answer any questions.
Great. Thanks. That was fantastic. We do have time for some questions. Anybody? Okay, Chris, can you hit?
Thanks. So presumably alpha-1 antitrypsin doesn't have the same effect. It's only the C-terminal peptide there. So what generates that peptide?
So
what's its mechanism? What does it bind to or inhibit or block?
So in the setting of pregnancy, there's a serine protease called HTRA1, so high temperature resistant protein 1 that's on the placenta. And in the placenta, there's also a fair amount of alpha-1 antitrypsin, and we think the increase of this serine protease on the placenta generates this fragment in cord blood. Because if you knock out HTRA1 in mice, their neutrophils are net competent, so they can make nets early on in life. In the setting of infection or inflammation, any serine protease that interacts with alpha-1 antitrypsin should, in general, cleave this fragment. And we're not exactly sure how many amino acid residues you have to have. We've toyed around with that a little bit, but we don't see it at a systemic level in any adult diseases that we've ever studied. We've done a little bit with human trauma patients, and we have seen it there. And I think you just have to have a ton of systemic serine protease activation to see this fragment. But I think locally, where you have platelets and neutrophils and coagulation cascades activating, like thrombin and other serine proteases, you probably generate this fragment as a negative feedback system. So you form nets, you get activated platelets, you form nets, you form thrombin, and you generate this fragment, and then it stops locally anything that's going on from a neutrophil extruding nets. But we're pretty sure that it's HTRA1 in the setting of pregnancy, but in terms of other in vivo reactions, we're not 100% sure what's generated.
But it's quite far from the reactive loop, though, right?
Say that again, sorry.
It's quite far from the reactive loop. So why would the serine proteases be cleaving that? And also, you'd have a P1 primes a proline on the peptide that you showed, which is a very unusual and very hard to cut residue.
Yeah, so it's a good question. I don't know the answer to it. I know, like, if you take alpha-1 antitrypsin and you put neutrophil elastase there, it will generate this fragment, and that fragment's capable of blocking net formation. So it seems that any serine protease that is there is capable of doing it. And I think I could be wrong. I'm more interested in the net inhibitory properties and how it's generated. But I thought it was near I thought it was near the center, like the inhibitory molecule of alpha-1 antitrypsin. I could be wrong.
I'll ask a naive question. So does aspirin interfere with net formation?
No, not that we know of. I mean, there is some evidence that aspirin can, obviously block platelet production or platelet function, but in our, I think if you have aspirin, that's an interesting question because I have another story that I could tell that shows that in some case aspirin works, in some other cases it doesn't work, but it's dependent on the level of platelet activation. So I think there's some threshold that you have to reach, and if you can block that threshold with aspirin, then you're good. But in some individuals where they have hyperreactive platelets, that aspirin can dampen the effect, but you're still above that threshold. So therefore, you still get platelet activation, and you still get neutrophil recruitment. You still get net release. Aspirin doesn't affect procoagulant platelet formation, so aspirinated people can still make PS on their surface. But there's some level of threshold that you have to reach, and if aspirin can drop it below that, then yes. But if I would say for a large portion of the population where their platelets may tend to be more hyperreactive, aspirin dampens it, but it doesn't block it.
And what are the properties of the HMGB1 deficient platelets? Are they altered in any other way other than the net formation?
So they do have some, I believe there's some alpha granule defects as well. From what I remember, Mackie Neal and the guys at Pittsburgh have designed those mice, and they're protected in venous thrombosis. They're protected in this arterial thrombosis, and I think they have a subtle alpha granule defect.
Just one quickie. You're talking about this peptide, NEF. Is it cleavable by plasma itself, the peptide itself?
I don't know the answer to that. No.
I only ask because it's not unlikely that this would be an adjunct therapy to TPA, possibly.
Yes. Yeah.
Right?
No, that's very true.
Thanks for that whirlwind tour. I'll just unloosen my seatbelt now. In your IFI-TM3 story, you showed a slide where some of the cells were infectable with dengue. And I was wondering, maybe those cells had a receptor mechanism, but others didn't. Now we know there's at least 2 receptors that dengue is entering. Maybe those cells were special.
Yeah, no, I think it's a great question, and it's something that, it's entirely possible. It's just something we haven't necessarily looked at. We know that the MEGs will make interferon. So we think that the reason why, from a hypothesis perspective, we think that some MEGs get infected for some reason, maybe receptor, maybe they just see the virus first. They induce interferon. They induce this type 1 interferon response, and that sends out signals to increase the translational response of IFI-TM3 and these surrounding cells, which can then protect them. But it is possible, and potentially even likely, that MEGs change their surface repertoire as they develop, and that at some early stage MEGs or late stage MEGs, they may have some receptors that are more prone to binding viruses, and those MEGs are being infected, and that's why there's this differential. It's just something we haven't looked at.
And there's a bunch of people, just keep seeing to whether my microphone's. There's a bunch of people here who are loading autologous platelets with nanoparticles loaded with special mRNAs. Do we see any potential for IFI-TM3 loading of platelets and using those as therapeutics in infection?
Yeah, I think it's an interesting idea to have IFI-TM3 there and potentially, blocking the systemic viremia or the systemic viral infection. I think, and especially in cases like in dengue and even in influenza, where you see thrombocytopenia, and you may not generate enough platelets that have IFI-TM on their surface, or you may not generate enough of an interferon response to fight the infection. If you had platelets that had IFI-TM3 that you could transfuse into a patient, they may do better off and being able to fight off the infection.
Thank you.
Thanks, Ed, for taking my question there. That was going to be for our later meeting. I just wanted to ask to clarify on that. for that IFIMT3 pep or protein. Do you know if it's endogenously translated in platelets in some sort of signal-dependent translation pathway, or is it something that's highly specific to the megakaryocytes?
So we know that it's mediated through STAT3 transcription and mTOR translation. We think that for the most part that it gets made in the megakaryocyte and then shuttled into the platelet. That doesn't necessarily mean that the platelet isn't capable of translating it. It's something that we haven't necessarily looked at before. But I mean, we do know in the setting of sepsis that platelets make more proteins, that they have increased translation based on S35 methionine studies. So in theory, it's very possible that the platelets are making some protein that I think, from my perspective, the amount of protein that they make is small. It may not have a systemic effect, but I think certainly at a local level, it could have a big effect.
Do you know the several what's the word, SNPs, variants of IFIT M3, and one or two of them are associated with worsened outcomes in COVID. Do they affect the clathrin? Are those sites, SNPs affect clathrin binding?
Yeah, that's a great question. We looked at that in the dengue story, and we saw that one of the SNPs that I can't remember off the top of my head, where they have it causes a truncated version. They were less likely to be protected in viral infections. So if you had the SNP in these megakaryocytes, they were more readily infected by dengue. We haven't looked at that in the setting of clathrin-mediated endocytosis.
But the site of it, is that where the SNPs are? Is that where the clathrin binding the Y?
Oh, I do not believe it is, but I don't remember off the top of my head. But that's a good question.
Good mechanism, right? If it is,
no, that's a very good question, and I just don't remember.
Very great talk. I have a question about IFITM3 knockout platelets in vitro aggregation. Do they have less aggregation in response to a wide range of agonist concentration, or is it just a low concentration?
Yeah, that's a great question. So it seems that there's a sensitivity. So at the lower concentrations, we were able to tease out this difference in aggregation, but at the higher, like thrombin, we didn't see an effect, and then at the higher levels of collagen and higher levels of 2-mes ADP, you begin to lose that phenotype.
All right. Years and years ago, not so many years ago, when I first was introduced to Robbie by Alyssa Wahlberg, and I can remember sitting there, and Robbie came in and she says, "That guy's going to be a star." And I guess she was right. He's done great, and we're really pleased that he came. So join me in thanking him. Don't forget about the party.